And in fact, this is a perfect, you can say, the start of this lecture that, we talk about this computing, we talk about this whole online teaching and other things. And you can understand that how sometimes, even for us, it can be a little confusing. So the theme of this, the workshop, which is sort of the human-centered computing, is the development of these technologies should take into account of that this the diversity of the people who's going to use it and all those things. So we will come to this part. So when I was approached to give this talk, I was a little bit confused because the theme is sort of the human-centered computing. And, if you try to understand the meaning of it, of course, it has a very wide scope. But I also try to see that how I can connect my talk with this, the actual intended theme with this, the human-centered computing. And since my sort of the area is primarily these days, I work a lot on the natural language processing. So what I decided that for the initial part of my talk, I will touch upon a little bit on the human-centered computing. What do we mean by human-centered computing, and what is sort of the definition. I will also try to give a bit more, through examples. I mean, nothing, let's say that in details, but then throwing some light on that, while developing, let's say that the technologies which uses NLP, what kind of issues we should be aware of to really, in true sense, we can use the scope or the concept of the human-centered computing towards our the NLP. So that's what I want to touch upon. So making sure that we don't diverge too much from the intended theme of the workshop. And then I will try to focus more on the task, or let's say where I am much more comfortable, or where my primary areas are there. So these two applications, and I will sort of slightly try to discuss also, or try to highlight a few points that in how these two applications, you can say that somehow loosely connected with the human-centered computing, or some of the scopes which has been sort of defined under the human-centered computing is sort of there. So that's the kind of an idea. And since the people are from the diverse background, what I understood from the organizers, and now the name of the organizers came. So sorry, I sort of did not really thank you. So I really thank the organizers for inviting me to give this talk. So let's now begin the real discussion. So let's start with touching upon a little bit on the human-centered computing. So as we see that the computing is impacting in all aspects of our life, perhaps the way we are interacting with each other, the way we communicate, the way we learn, and people sort of say that one of the kind of good point, or the positives of this COVID pandemic, which is still going on, is that we now understood, or we try to sort of perhaps take the advantage of the technology which is available to us. Before this COVID, I think we would be seeing each other face to face. Of course, it has its own benefit, but then at the same time, it also has a lot of other constraints issues. So we see that this computing, the technologies allow us, the way where how we learn, how we interact with each other, how we communicate, how we work, and so on. However, the most of the development happens See, for example, the tool which is being developed for the teaching purpose, for example, did not really look into the all other aspects. And again, I want to sort of highlight the issue of, let's say, for example, during this COVID pandemic, when the classes started going online, people, initially people thought that, oh, we can, teach the classes online. But then at the same time, perhaps we have to also deal with the issue that how to really keep students engaged and so many other aspects of learning. It's not that just you create your slides or the video and you just send to the students. So, but then while the technologies which are being initially developed, they may not have really thought about these aspects. And only during this whole the pandemic situation, perhaps all people who are working on those areas started looking into these aspects as well. So, the point which I want to make here is that we keep developing new technologies. There are a lot of things, but then the huge portion of these developed technologies are unfriendly, unnatural, in the sense that you may not always have the people who are, let's say, that the experts in using these technologies. So, for example, if people are not accustomed to using this kind of video calls or taking this online exams or taking the exams on the mobile, so these technology you can of course develop, but then whether the end user who is right now, for example, in the current situation is at home, and if their parents are not well aware of these technologies, they face a lot of issues. So, in one hand, we say that these technologies is being developed to sort of, empower people, help people. But on the other hand, we say that these technologies are perhaps helping only them who can really handle them, who really understands them to certain extent, or who are accustomed to using these things, but not to the people who are, let's say, that not so well versed with these things. So you can say that the computing technologies, of course, is a gateways or bringing opportunities and so on. But then at the same time, it also creates a barrier, because there are a lot of people who cannot use it, and then they are there is a sort of the huge risk of a good chunk of people being left out. And again, when I talk about the HCC perspective, the human-centered perspective from the NLP point of view, and then I will try to show there some examples. So right now I'm only talking about in terms of, let's say, that the abstract concepts, but then I will show you some specific examples that how some of these technologies actually creates a problem, and then we should be aware of. And that is where the scope of HCC, or the human-centered computing, comes. That you cannot just keep continuing developing the new technologies in isolation. You cannot keep developing these ones without, let's say, that taking care of the diverse kind of issues in the human abilities and their limitations, the social and the cultural, the biases or the awareness, and how, let's say, that across the different the individuals, across the different age groups, different the social and cultural backgrounds, or under certain specific situations, can actually adapt to this one. So the scope of HCC is that we need to bring a paradigm shift in our development of the computing technologies. So where our focus is not only in the development of the technology, but also we need to take account of these aspects which I have just mentioned. So you cannot just say that I want to just develop the video calling technology. No. You have to also say that you need to develop these technologies where, for example, especially in the Indian context, where the network connections are not good, where you have the intermediate connection issues, people uses mostly with the mobile, they are going to access these things and so on. So you cannot assume that everyone has a broadband fiber connection and then the kind of a good bandwidth to really handle there's lots of data coming and going. And then you also need to sort of understand that there will be many houses where people may not be aware of these things. They will not be able to handle them, or often, let's say that if you are talking about the children, their parents may not be at home and they are with the grandparents, and then they may not be so accustomed to using of these technologies. So you need to sort of when we talk about the developing technologies, you need to really take care of these aspects. So that is why we say that while you develop in the applications, you need to take the input from the diverse sources. You cannot just say that, okay, you have one collection of the people and for them you are going to sort of try to develop these technologies. You may have to sort of look into the diverse sources. And also, while communicating that, okay, what you have developed and how to use them, you cannot just say how to in only one language. Again, for example, in India, you may have to sort of translate in everything into the vernacular languages so that people can understand that what is there if they are not, let's say, that knowing English or Hindi, maybe, for example. Yeah, so these points have sort of, already I have highlighted. So, of course, there are a lot of definition which is there. I picked this one because then it would be easy for me to relate my subsequent talk in the context of the human-centered computing. So the Hoffman, who defines that the development, the evaluation, and dissemination of the technology that is intended to amplify and extend the human capabilities to perceive, understand, reason, decide, and collaborate. So, any kind of a technology which we are going to develop, which needs to sort of amplify the human capabilities in sort of, the perceiving the information, understanding what is there, to reason about the data or all the information which has been sort of given, and then take a kind of a decision and so on. And that can also sort of lead to the, the achieve, maintain, and exercise expertise. So you can see that the talk, the other aspect which I'm going to, talk today is sort of trying to look more into the first point where we try to develop, let's say, that the machine learning based methods in the biomedical domain with this one objective of to sort of the amplify and then extend our capabilities to understand a lot of the different kind of the data which is there, and in turn those things should help us to for example treat the disease better, or to come up with the drugs which can be sort of helpful not only for common diseases but also the rare diseases, or this kind of a the sudden pandemic kind of a situation which comes in and so on. So so that is where the other part of the talk will focus. So this is all I wanted to say about the human-centered computing. And now I will focus on that how in NLP we also see certain kind of the issues, and that needs to be again handled within the scope of the human-centered computing. And the premise of this is that in today's, let's say, that the natural language processing task, what you see is when we talk about the natural language, it's basically the languages which you see in the text whether it can be, let's say that the English, it can be Hindi, it can be German, it can be Sanskrit, it can be Assamese, it can be Bengali, and so on. So, and if with the huge collection of those text data is available to you, how do you do a lot of the automation and the processing? In those cases, what we do is to use the machine learning based method. We need to represent each of the words in any given language in terms of the real vector space. So that is what is called embeddings. And mostly nowadays, the neural network models, which are very common. So these are very, very common, and then the vector representation for each of the words, we learn about it. So for it, I can just sort of mention. So if there is any word, for example, even the NLP is a one, let's say that the word which we have, and what we want to do is that we want to present them into a d-dimensional vector space. Okay, so we want to present them into, so that is what the embedding says. Okay, d-dimensional vector space. So this would mean that this would be equal to something like 0.3, comma 2.4, comma 5.1. I'm just sort of randomly writing something here to just give you an idea that what I mean by embedding. So let's say that if the D is 4, if the D is equal to 4, then each of the word can be represented into the 4-dimensional vector. Okay, so this is just one example here. So that is an embedding. So now how we obtain these embeddings? We obtain these embeddings by, let's say that developing an algorithm which looks into the huge collection of the text, and these embeddings also represent, or it is expected them to represent the true meaning of those words. So if you have a particular representation for the word like bad, okay, then it should also if you look into the nearest vector corresponding to the bad, then all, let's say that the poor or synonyms for the bad should be there. So the word like the good and its vector representation should be far away from the vector's representation for the word bad. And that is being learned again through the large collection of the text. Now, what happens is that while you learning these representation, they actually imbibes or takes that kind of a biases which is present in the text. So all the the societal bias in the data which is present there, that is being sort of incorporated into this, and then if you use those models for certain applications, and then it might have the adverse impact. And again, I will sort of show that what kind of an adverse impact it will have. So I will try to again explain through this example. So let's look into this one example. Now, as we are aware that today's time, the social media is very active, be it WhatsApp, be it Twitter, be it Facebook, and then there are many more such social media platforms exist. And then there also we know that how people try to bully each other, and that how the hate speech is being going on. So there's a lot of interest in the NLP community. So can we detect, or these platform which is being developed for like the Twitter, Facebook, their engineers, their computer scientists are actually looking into this aspect, that can they automatically detect, because there the millions and the billions of those messages are being sort of posted, so it cannot be manually checked. So of course you need to develop some automatic technique to identify those hate speech and so on. Can we do it automatically? And if you try to do this automatically, then you have to be aware that some of the words which could be the indicator of a hate speech, but then some of those words could also be a kind of a the slang word, or very, you can say that the people in a particular community, those words are used without having any meaning, or even in a very friendly manner. So if you emphasize about on those words, or if your data is collected in such a manner that those kind of words are being sort of being specified as a strong indicator of the hate speech, then it might adversely impact certain people from this certain dialect. So this is one example that on the left hand side you see some of the messages are there, and then you have this automatic method which is actually try to detect that whether it's a hate speech or not, and then what kind of confidence you can have. And what you see is that there are people, when a particular community, people from the community try to sort of, in a very friendly manner talk about something, their perceived toxicity level is very high, although it's not the toxic or the hate speech. So this sort of indicates that while developing this kind of an automatic method, you need to take care of the kind of the social and the cultural, the aspects of the people, which how they use their language and so on, and then only you can sort of develop a good model. Similarly, let's say that there are some unintended biases towards the certain identity term. So, for example, people might use the gay and the lesbian as a kind of again, in hate speech, people can use this kind of terms, but not always these terms are actually means that somebody is talking in having the hate in its speech or its conversation. So again, but then what happens is that this kind of a thing is also gets high score, because your data, what you are using in to train your model, is actually whenever in your training data, whenever you have seen these words, it's basically had the malintention or trying to insult people in some sense. So that kind of a biases which are present there, and that leads to some incorrect score being assigned to the things which should not be. And similarly, if you see the kind of a data which is sort of taking, it might sort of again incorrectly assign these kind of words which are appearing the high toxicity. So, which is sort of the incorrect. So that is why again we need to bring the aspects of we need to sort of make certain changes in the models which can understand these aspects better. And then these are again the further examples where if you have certain disabilities, and that again reflected as a high toxicity score, whereas in that particular context, it's not actually the hate speech or the toxic statement. Again, the reason is that the data which you had is actually the bias towards having these kind of words being used more for the hate speech and so on. And then we see when you have these word representation, you can try to sort of have the prediction when you try to say that he is, and then try to say that what it will the next word would be, or you can give it as a kind of an analogy task. And what people have observed that it actually, this kind of stereotypes we have in our society often, that the men are basically the one who is working, working in the sense of, let's say that they are office goers, they are engineers, they are doctors, and so on. But when we see in our society, perhaps that when we talk about women, it's more towards that they are housewife or homemaker and so on. So, and that also being reflected in your data, and that kind of the information is sort of taken into your representation as well. So, so again, we need to sort of make sure that this kind of a biases should be properly taken care of. You can ask me this question, that if our society is having these biases, then how can we, let's say that, say our models to sort of not to reflect that. And then should not we then just leave it to them, because leave like as it is, because this is what our society has, this kind of a notion, and then model just takes from takes the data which is coming from the society, and then model is just learning that. So the question is that if we allow to do that, then that is going to harm certain groups of people. There's a difference, like when in the society, when we have this kind of let's say that the biases present in the society, but when it comes to the certain kind of an evaluation which is being done manually, we take a lot of other information as well. There's a lot of, let's say that the contextual information is being taken into account, but that may not be feasible when you want to do it automatically. And that is why it might harm certain group of people. So here is an example that, when let's say that Amazon has this, its resume sorting things, when they sort of, see that there's the female candidate, it might try to sort of penalize those resumes, or let's say that try to not to select that. So that could be, and again, the issue. But then if, let's say that if it is being done manually, then it would not be the similar kind of issues. So that is why the automatic way of using this technology which is being developed to do something might have, create an unintended harm to certain group of people. So, so some kind of an examples of this harm is the allocative harm, where this automatic system try to allocate or withhold a certain opportunity or resources to a certain group of people. So, for example, this resume selection thing, that as soon as you perhaps see that the candidate is female, you might want to say that, oh, females are mostly intended towards the homemakers, so they cannot be a right candidate, and so on. So you have the bias associated with your data, and this automatic system will reject or relatively, give the poor score to that resume. Or associative harm. So when, let's say, that your automatic system tries to reinforce some kind of the information which is related to a particular group of people. So as we have talked about the cases of the hate speech, the racial biases, and so on. So there, or a certain kind of disability and so on. So then you try to reinforce those kind of the notion by by going through your data only, and then not sort of taking care of that. Okay, this cultural group might have certain different thing, and you should not really penalize in that way. So that kind of an associative harm would be there. So there is, these are some of the examples which I have tried to highlight here. But then there are n number of similar kind of examples. You can find it in the vision domain. Also, when we talk about these face recognition system and all those things, there are a lot of hue and cry about those systems as well, where they always try to sort of associate, let's say, that relatively black or non-whitish face to a criminal and so on. So this kind of an issue is there, and people have studied a lot on these aspects, that what kind of biases which are present in the data, which gives rise this kind of an issue, and how this can be taken into account, and so on. So these are some of the, if you are interested into these aspects, maybe you can start looking into some of these papers which actually talk about, specifically in the NLP domain, it actually talks about some of these issues and how to sort of take care of. So for example, one of the first paper which says that how to debias your word embeddings or the vector representation of the words to make sure that it does not make this kind of a mistake, or this kind of a stereotyping. And many of these slides are actually coming from this. If you go to this website, there are good resources are available, and as well as the slides and then the papers and the talks. You can look into that. So this is a good place to look at it, the relevant resources related to the fairness research in NLP. But then this, as I say, is very much or very closely related to the human-centered computing in NLP domain. So that is about I wanted to sort of connect with this theme. Now, the next part of the talk is sort of talking about the two different applications, where in one case we are trying to understand the natural language in the biomedical domain, and we use again the machine learning methods or statistical methods to do that. And in the second case, we look into the other kind of a language, what I call is the genomic language, and how we can use machine learning methods to decipher or enhance our understanding of genomic complexities. So the first application where I am going to talk about this biomedical domain, here the intention is that how let's say that the kind of the methods we are going to develop is going to helpful to the doctors to retrieve, let's say that, useful information from a large collection of the data. So in this particular application, I have talked about electronic health record, but then you can do the similar things from the biomedical literatures and so on. So let's talk about that, how with the large collection of the electronic health record, what kind of an information can be sort of automatically mined, and which can be perhaps helpful to the clinicians or the doctors to understand some of the uncommon link among, let's say that, that you have a large collection of the text, and what kind of a method you should look into, and how this you look into this, the large collection of the text, and automatically extract out the relevant information, and after extracting out the relevant information, make, let's say that, some kind of a knowledge which would be useful for the medical practitioners. But having this information in this kind of a plain text format is not going to be easy to handle, even with the the automatic methods, the ML methods. So what you want to do is that you want to convert these text data into some sort of a graph like this, and this graph could be, again, if instead of, if you have let's say that a lot of this kind of a graph, then it would be easier to perhaps find out how strong this kind of a relation between the two disease nodes are, or what kind of a relation is between the medicine and the disease, so how strong that is, and so on. So that you can do once you have the lot of this kind of a graph extracted out from this kind of electronic health record or discharge summary. I mean, similarly, you can do the same thing with respect to the biomedical literatures. So what we did is that we try to look into one specific kind of a problem. What we did is that we try to sort of infer rules to indicate how presence of one disease or a disorder can influence the onset of some other disease. So basically, we try to identify the comorbid diseases, or the diseases which could be a risk factor for other diseases. That's what we wanted to identify, and we made, let's say that, a kind of a one small flow chart that how we can do that. And this is what is that you have the electronic health record, a large collection of it, and then you try to sort of identify from these electronic health record that what kind of the diseases are present, and among those diseases, what kind of relations are there, and then you might want to sort of apply. So this is for one electronic health record, but if you have several thousands, 10,000s, or the 100,000s or millions of such records, then you can, for each one of them, you extract that information, you put them into this matrix, then you try to perhaps use some statistical method or machine learning methods to identify which one is the more the strong relations, and then you can perhaps try to do some validation either through the literature or through the genomic level validations, and after you have done those validations, you can give certain rules out of that. So that's a kind of a the flow chart. It's a generic flow chart. You can change that, what kind of a methods which you want to use, and what kind of a validation you may want to use to confer or come up with the final valid rules. So again, I'm just trying to highlight the steps, the important steps which you are required, and that those are not, and let's say that easy steps to really work with, or still these are the challenges. So first, one of the challenges which we face is that when you have these the text, the disease names may not be present in just one single way. Because, sorry, is there any question? Is there any question, or should I continue?
No, sir. You can continue, sir.
Oh, okay. So, yeah. So the one of the first challenges is that the disease names do not just appear in a standard way, because these discharge summaries was written by the different, let's say, the doctors or the nurses or paramedical staffs, and they may not use always the standard full name of the disease. They might use a lot of different abbreviation, or they have their own way to sort of refer to a particular condition, and they might write in that way. So the first thing is that you need to identify those names, and then those variants of those names again need to be mapped to a single kind of a canonical form. So that's the one challenge that you want to do. The second challenge is that it may not be only with this some minor variation in their name, but it could be completely different name for the same disease, and that would be again need to be mapped to the canonical name of the disease. So this is the two level of the normalizations you have to do it now. But before that, how we are going to identify those disease names? So it cannot be possible, entirely possible, by looking into the dictionary, or let's say that the well-established thesaurus of the medical thesaurus. It may not be able to include all varieties of the disease names. So what we do is that we try to look for some models which can try to do this for us to identify the disease names, and which often in the NLP terminologies is named as a named entity recognition task. So you have this kind of a text which is there, and you want to sort of identify all types of the disease names which are present there, what all types of the test names which are present there, and all types of the treatments which are given, whether in terms of the medicine, whether in terms of some other things. So all of these comes as a treatment. So you want to perhaps identify all of these different types of the entities. And what we can do is that we formulate it as a supervised sequence labeling problem. Sequence labeling problem means is that you want to tag each word in the given sequence. So this particular word is not of the entity of the interest, so it will be labeled as other. This is also not, so this would be also labeled as other. Now this is the one which is sort of seems like a treatment, so this would be identified as a treatment. This is the disease or the problem or the disorder. So now you see here that I have used something like the So this is something called the BIO. In the sense, the objective here is when you have disease names which span more than one word. So as you can see, the congestive heart failure is 3 words. So this entire, all 3 words together indicates the problem. So I need to identify that, okay, this disease name starts from here and it continues. So these are the intermediates. And then as soon as I see the other, then I know that what is the boundary of it. So I need to do a little bit of the post-processing, but then I would know that exact the boundary of this particular disease name. Apart from BIO, there are a lot of other tagging schemes, but let's keep it simple, and we can do this the BIO kind of a tagging. Okay. So as I said that we want to convert this the name entity recognition as a supervised sequence learning problem, where you have a training data. In each of the training sample has a sequence of words, and for each sequence you have the corresponding labels, and so and then each of them are of the varying length. So that is why this is a X and I. Okay, and then your objective is to find an approximation or hypothesis F which can map any of these SI to these the YIs or the TIs. The YI is basically the label sequence given by T1 to TNI. Okay, so this is what you want to do. So here is one the example of a neural network model that we have the sentence given to us, and then what we do is that we obtain its the embeddings for each of the word. First we have obtained the word embedding, then corresponding to these words we are also obtaining the character embedding. So character based sort of the representation. I will sort of talk in the next slide what I meant. And I also use, for example, the part of speech tag information. So if something is a noun, something is a verb, so part of the speech implies that. So for each part of speech, I have, let's say that again, some dimensional representation. So it could be 5 or 10, depending on the my choice. So all of these representations are concatenated together. So if let's say that my word embedding is of the dimension 100, character embedding is of the dimension, let's say 20, and the part of speech embedding dimension is 10. So it would be 100 plus 20 plus 10. So each of the word would be represented by 130 dimension. Now these are the hyperparameters that we can sort of tune. But for this discussion, let's assume that you have fixed that and you obtain a representation there. And then you feed them into the neural network model. Again, I'm not going into the details of these models. I'm just giving you an architecture highlight. But if you are interested, let me know. I can share the papers with you, and then you can understand that one. And you have the final, the final layer where you can have the prediction. So you see, why we call it the domain invariant model. The reason is that initially it was the initial models, so the previous models were more dependent on that. How do you define the different kind of features? And then you use some machine learning methods. Often, of course, the neural network can also be used, but then it was mostly some other methods like hidden Markov model or maximum entropy Markov model and so on. So those different models are being used. The problem was that defining the feature was a challenge, and if you want to use those models, you won't be able to replicate the result because those feature informations are given in such a way that you won't be able to really replicate that, and the codes are also not often available. And then, if you change your domain from, let's say, that the clinical text versus, let's say, that the life science research article, you may have to define the features again, and then you accordingly retrain your model. But in this context, we wanted this model to be the domain invariant, whether we talk about the clinical text, whether we talk about general domain text, whether we talk about the life science research articles. In each of these cases, we should be able to use the same model without changing anything here. Only thing will change is perhaps the corresponding data set. So that is why this name is domain invariant model. And in the character embedding, the idea was that, often, specifically in the clinical domain, there are certain clues are present in the words. So some words which are ending with, certain suffix indicates that this could be a disease, or a certain word which indicates have some kind of a prefix, then it means something very special. So to import or get those knowledge or information learned automatically, we wanted to have a character-based embedding representation here. So you have, for each of this character, again, you have certain dimension representation. And again, you use some neural model to train that. And the final representation you try to obtain from there. Once you get these representation, you concatenate them and combine them together to obtain a single representation for the entire word. So, so that has been sort of incorporated here to obtain this character embedding. Now, so this is what you wanted to do, but whether this actually does the intended task, that we can check by looking into the some random word and then see that whether it is able to pick certain kind of information which we wanted to do. So, for example, the synergistic. Now, if you use this character-based embedding, then you see that it is able to capture this notion of that when this thick word is sort of coming. So it is trying to sort of say that these words are very similar to this one. And then similarly here, the this, which is sort of there. So this prefix information is somehow getting learned, and then accordingly the nearest words to it is coming there. But if you but then again you can see that this character-based embedding try to give more importance to the the structure or morphological structure of words. So that may not always be the ideal thing you want, and it can easily be misled. So that is why you also want to have the word embedding present there, because that takes the more generic context information. So by looking into this table, you can sort of see that. So for example, the dysfunction, the impairment, the impaired, or the disturbances, these are also related one. But then these words are, of course, having a similar meaning, associated with this the original word or the source word. But then none of them were able to sort of pick the this kind of a prefix or the morphological structure of the word. But if you use the character-based embedding, then you might be able to obtain that. So that's the point which we want to highlight here. So both of them using simultaneously is going to be helpful, and that is what we have found, that compared to the other models, if we use the word embedding, the character-based embedding, as well as the part of speech embedding, it actually helps improving the result. And so that is about the entity recognition, and then you can also do the relation extraction from there. And this is what again with the same example, you see that we want to identify that whether some test revealed some problem or some treatment actually helped as in let's say that recovering from that problem or not. So that kind of relation which we want to sort of identify. So you just recall that the first graph I have shown you, we wanted to extract this kind of information. So that is what we are doing here, that we given this kind of a text, we want to identify this kind of a relation as our first step, and then once you convert this text into this kind of a relational graph, then you can again mine from several such relational graph to get more useful information. You can also identify not only this kind of information, but you can also identify the drug-drug interaction information and so on. What we did is that, of course, these are about identifying the relations, predefined relations, and earlier we have shown that how to identify the disease names. Based on this, what we did is that we also try to, you have the labeled diseases, and you have also seen that these diseases were, whether they were found during the current admission of the patient or whether they were the present, or whether they were the part of their the family history. So all of this is sort of been taken into this information has been taken into from the discharge summary, and we hypothesize that okay, this kind of a pair could be a comorbid pair, or this could be a causal pair, and so on, or risk factor. And then we wanted to sort of, using the simple chi-square test, to identify that whether they are really significant based on with what frequencies this kind of a pair appears in as a comorbid or causal pair. So when we did that, we were able to identify certain diseases which could be caused, can be said as that they are the comorbid pairs, or they are a causal pair. So once we have identified that, then what we do is that we try to sort of validate through literature. And what we do here is that we take the disease names and what kind of, let's say, the relation which we are looking at it. So if we are looking at the comorbid relations, so we try to represent that comorbid relation with several synonymous phrases, and then try to sort of extract out from the database of life science articles that, and how many articles this has been sort of mentioned as a causal or risk factor. And we have taken all those articles which actually having this kind of an the informations, and we look into these abstracts, and then we look into this the patterns, and extracted out that all of them, the out of whatever we have able to extract, how many of them is actually talking about that particular pair in that particular for that particular relation. So this kind of an information. So if this is a high percentage, so that we think that is a kind of a indirect validation to our the results that yes, that whatever we are trying to extract out automatically through only going through these the discharge summaries is actually correct. What we can also do is that we can also look into the genomic data, where what we do is that we look into the different the networks of, let's say, that the protein-protein networks, interaction networks, or gene regulatory networks, and then see that whether, if I'm saying that these two diseases are related, whether the known the genes or the known pathways or the pathways which are sort of associated with these diseases are actually somehow interlinked in our network. And so this is what we try to sort of look into, is that, okay, you have a subnetwork of genes associated with disease 1, some networks of genes of disease 2, and then you want to sort of do this, perform the network analysis to identify the proximity of these genes to these genes, or you can do that whether they are part of certain the pathways which can crosstalk, or you can do several kind of analysis to generate lot of hypotheses which can further validated through the proper experiment. So I'm not claiming here is that you can directly say that, okay, this is what is actually happening. All automatic methods are basically just helping the biologists to identify or reduce their hypothesis space or helping them to prioritize among several hypotheses which could be tested to understand, for example, the relation between these two diseases can be sort of prioritized based on this kind of information, that, okay, whether some of the genes associated with this disease is very closely to the other disease, or what are the pathways which are associated with this disease 1, and what are the pathways which are associated with disease 2, and do we see that they are sort of crossing each other into the larger network. So those things can help them to further prioritize their the experiments to help them better understand the different diseases. So we try to do certain basic analysis here, and we did find that, okay, there are certain relations which we can identify. So this is one kind of an example, or one kind of a work which we can do to help out. Again, you can see that you can help out to the doctors, to the biologists who actually work with these diseases to better understand those diseases, or come up with the new novel drug and so on. So in that aspect, this is sort of related to the human-centered computing. As I said, that it can help us to better understand the large collection of the data, and from there we can generate certain hypotheses which in turn can help the biologists, the medical professionals to better understand the disease and so on. We can also do the traditional, we can also help in coming up with the uses of existing drug for some rare disease or some novel disease. So as you might have, if you have followed through this whole COVID pandemic, we have seen that there's a lot of initial days, people try to come up with a lot of the existing drug which can be used for the COVID patients. Some drug had, some limited success as well. So that is what is called a drug repurposing. Those existing drugs were not designed or not intended in its original way to be used for the COVID patients. But what people have realized that, oh, under certain conditions or in a specific kind of cases, those drugs can be given to the COVID patients as well. So that is what is called a drug repurposing. You are repurposing the existing drug for some other disease. So that is what the drug repurposing means, is that use of the approved drugs or a drug under development for a different indication than the original target. Okay, and there are a lot of the other names are there. And now this drug repurposing can be done into the smaller amount of the time, as it sort of compares with the traditional drug discovery versus the drug repurposing. And you can see that in recent times, a lot of drugs has been approved by FDA, which is basically the authority in US to sort of say that which drugs can be used. So that with that, about 30% of the approved drugs in the last 13 years is basically coming from the drug repurposing. And this drug repurposing is also useful for, as I said, that for the rare diseases where the pharmaceutical companies have not much economical interest to really invest a lot, because those rare diseases are very few people are getting affected by those diseases, and they will not be able to recover the cost. But then if you do the repurposing of the drug, then it would be really helpful for people going through this rare diseases, or for the novel diseases like the COVID situation. So there are several such success stories where the drug which is sort of designed for original indication, and then it started people using it for different indication. So that is the basic idea, but then how we can formulate a problem, as an algorithmic problem or computational problem. So the idea here of the drug repurposing is that you have certain information available in the drug space, and based on, let's say, that the drug's chemical composition, structure, or its toxicity levels, or some other information which is available in the literature, you know that these two drugs are similar to each other and how they are related and so on. So this is the one space you have. Then you have the disease space, where you have, again, the similar kind of an information, how the two diseases are similar, they belong to the similar family, and so on. So you have this kind of the graph which is available to you. And then you might have some other information related to that, how certain drugs, of course, some drugs are used for this particular disease, so that is a direct association. Or they have, so these are kind of a direct association, and then your objective is to find out that whether this drug can be used for this disease, because these two diseases have something in common, or they are somehow related based on either their phenotypic characteristics, based on their the gene which is sort of associated with that, and that genes in somehow being attacked by that drug. So that might also help in this particular disease. So based on those notions, we can actually formulate this as a computational problem to design or come up with a novel indication for the existing drugs. Of course, we have done some work, but I will try to skip out. We don't have much time left out. So we have developed some model which helped us to identify some existing drug for Alzheimer's disease. And what we saw is that these drugs were somehow related to the neurodegenerative diseases, may not be for the Alzheimer disease, which is again, of course, related to the neurodegenerative disease, but uh, they are these drugs are not developed for the Alzheimer directly, but then we see that these can also be used. I mean, our models predicts that this can be used it for Alzheimer as well. And then when we look into the literature, there are certain candidates here, for example, this, which is actually under the clinical trials for treating Alzheimer disease and so on. So we get certain this kind of indications for our model. Of course, this can be further improved, but these are like some basic studies. We have done it. It can be further improved, of course. And then we have also looked into for hypertension and so on, and then similar kind of evidences. We have seen that some of the drugs which has been sort of used is, or our model says that can be used for hypertension, actually used for different type of the hypertension. So those things are being sort of detected in our model. So definitely, with some more information, some improvement in the model, we can perhaps come up with a better indication of the novel disease for existing drug. Now, let me quickly go through, because I have only 10 minutes almost left. So I will try to summarize that what we can do to understand, let's say, that some of the genomic complexities. So we are, what is happening is that, at one level, what we call is a genotype, which is basically our information in terms of the DNA, which is basically the genomic, which contains, let's say, that our genomic information. And then we have something which what we observe, the phenotype, certain characteristics of the diseases or the symptoms. So people, of course, one way to is that, you look into the genomic compositions of a person's or human beings, and then from there try to predict or understand that how it directly relates to the certain kind of phenotypes. Or what you can do is, so that is one way to look at it. The other way you can look into is that instead of directly looking into this, because this connection is very complex. The reason being is that our DNA. Let me see whether I have slides now. I don't have. So with DNA, it actually gets converted into the protein, or it actually, there's a lot of intermediate steps which actually give rise to the proteins, and those proteins is actually interact and have more functional, let's say, that the properties which can be directly related to the disease. So instead of going to direct prediction from genotype to phenotype, we may want to look into the genotype and look into these intermediate cell variables. And once we are able to understand these intermediate cell variables, that can help us to directly relate it with the certain phenotypes better. So with this objective, we try to focus on that. Can we, given, let's say, that the genomic information, we can understand some of the cell variables. So one of the cell variable is what we are looking into is something called splicing. The splicing is, let me try to explain here. So, we have genomic sequence which contains several thousands of the genes, and one particular gene has, the basic structure look like this one, that you have the DNA sequences. Of course, the genes is again the one segment of the DNA sequences, and it has certain functional regions. So one functional region is called the exome, and other one is the introne, and then these are sort of arranged in alternative fashion. So you have exon 1, then intron 1, exon 2, and intron 2, and so on. And then the first step is the transcription, where this gene gets converted into the mRNA. And so there, what happens is that if you only look into these exons, introns, and exons, then what happens is that these introns parts get spliced out, and the exons are joined together. Okay, so they are ligated together, and then this is what we call the mature transcript, and then this gets converted into protein, which becomes the functional part. So, but our interest is here, what we call the splicing, that how we can identify these splicing signals automatically. So people have sort of, after a lot of experiments and other things, people were able to understand that, okay, these are the structure of the exon and introns, and so on, and these are the, let's say that, the splicing sites. Now, our question was, can we, given let's say that this whole sequence, can we identify these the splicing sites? So this is my exon part, this is the intron part, this is again another exon, and what happens is that this intron parts get cut out here, and you can see that the place which it gets cut out starts from they have these two nucleotides, what we call is the guanine and thymine. So G and T, the symbol it was represented as G and T, and this is called a donor site, and this is called the acceptor site. So our idea was that, can we automatically detect these splicing sites given the entire, let's say that these exon intron segment. So identification of the splice junctions, can we reduce the dependency on the existing knowledge, and can we learn these signals automatically, that see, this is because of the presence of the GT and presence of the AZ here, this is the intronic region, and this should be sort of spliced out. Can we learn this automatically? Because of course now we know a lot about the splice sites, but at the same time we don't know a lot of other functionalities. So if we are able to show that we can understand, we can figure out this kind of features from splicing sites, then we can claim that we can do it for some other problem as well. So, so the first thing what we did is that we again, trying to borrow the information of the model which we have in case of the natural language, while dealing with the English text, that you have to have a word. So we took let's say that the k-mers, or the k consecutive characters of the nucleotides, or k consecutive nucleotides, and say that, okay, these constitutes a word, and for those words we try to give them a representation, and then try to learn a certain feature, and then use them as a binary classifier. So that's the idea here. But what we felt that if we use this kind of a model, so first of all, we don't have any particular justification to choose that why we should a length of 3 nucleotides as of one word, or length of 4 nucleotides as another word, and so on. So that we were not be able to understand, or not be able to sort of give a proper explanation to that. And the second question, which was the bigger question, that can we identify these the features which we wanted to extract out from the learned models, that these are the regions which are responsible, or which are the features. So since this model was not able to do that. We are unable to interpret the features, and there was no mean to explain the embedding space. So we looked into some other model, which is what we call is the interpretable BioLSTM network. And from there, of course, the idea was you have each of the characters, you use them directly, you don't constitute any words there. Then on top of it, you put one particular kind of a neural network. Then let them also learn that which part of these sort of the characters or the regions are important through this attention layer, and then try to build a classifier on top of that. So I'm sort of skipping some of the part, but let's try to see that when we do that, we can actually visualize the positive and the negative regions. So so the yellow ones are the one which are sort of the incorrect. Those are not the splice sites, and these are the splice sites. So you see that with this model, we were able to learn a representation for each of the splice sites, which are able to distinguish to a large extent. As you can see that all the two ones are clustered together. Each of these different samples. Of course, there are certain boundary regions where it will be confusing, but most of the other ones are definitely clearly separable. And that is also being reflected in terms of the performance. While our initial model gives only the 71 kind of a performance in that range, 71% accuracy or 71% F1 score, with this model we would be able to reach up to the 98%. And then our overall objective was can we extract out certain features which can be biologically relevant. So again, I'm skipping about these different methods with which we have tried to obtain that. But what we saw that yes, these were able to see that this is the one site of the splice site, what we call the donor site, and there is a peak of it. So it indicates that this is something of the significant region. Okay, and similarly in the acceptor also you see that there is a certain significant region. So all of these methods were able to sort of pick those cases, and this is what we call is the non-canonical cases. Is what we see that the non-canonical cases are there. Let me go back to. I will explain this and then I will stop here. So this is your what we see, the canonical sites are those where all the donor sites, the starting of the intron and the ending of the intron has intron has GT as a signal, and ending of the introns has AG as signal. So that is all of, if your all splice sites contains that, so those are called the canonical splice sites. But there are sites, the splice sites, which does not have these signals present in their respective donor and acceptor positions. So those are called non-canonical, and they have different kind of a mix signals, and that was also picked up by one of the method which says that it has, of course, some signal at this particular site, but then there are a lot of weak signal present. And that also sort of the literature people have discussed that in case of the non-canonical splicing, they have this kind of a variations present compared to the canonical cases. So, then we try to extract out a lot of these different kind of features which is already known with respect to the splicing sites, and some methods were able to do it better than the other methods, and that is what we wanted to sort of compare, that which one were able to give them the importance to the sequence position, which one were able to give importance to the donor site consensus or acceptor site consensus, or there are certain important points, something called the branch point. So those are like certain biological characteristics present in the genomic sequences, and our model was trying to sort of help out or extract out these informations, so that could be much more informative to, let's say, that to the biologists, instead of just saying that, see, my model has this much of accuracy. So you can say that this is again one example of where in sense that it's in some sense it's within the scope of human-centered computing, because I'm not just trying to develop a model which is for a particular domain, and just I give the prediction result and then I just say that, okay, my task is done. Rather, we are trying to also sit along with the biologist and then try to understand that for this kind of features, what could be the important features, or what kind of information it can be extracted out from the models which could be useful to them. So in that sense, you can say that these are the works which are actually related to, in some loose sense, it's related to the human-centered computing as well. So with this, I end my talk. I have taken slightly more than I was supposed to. Sorry for that. Now I am open to the questions. Now it's to Dr. Mehdi, to you. If there are any questions.
Sir. So participants, you can ask your questions.
Hello.
Yes,
hello.
Yes.
My query is regarding the meta-analysis and NLP actually. So is this a similar thing, or meta-analysis we can use the NLP?
Sorry, I did not really understand your question. So,
My query is regarding the meta-analysis in biomedical, and
okay.
what is the relation based on the NLP actually, natural language processing?
So, do you want to sort of reflect to the part of the talk which I have talked about, this how to infer from EHR some information?
Yes.
How
meta-analysis means it's like a statistical based models to analyze huge data from biomedical, like cancer, and combining everything, means all the data. So can we use this NLP there?
See again, in fact, the first part of my talk, the application talk, you can use, you can see the glimpse of that actually when we talked about, for example, drug repurposing problem.
Yes,
so there actually we use a lot of diverse kind of an information. It's not only the literature, it's also the different structured database which has let's say that, for example, drugs chemical composition information, drugs adverse, let's say that reactions, what are the drugs target information. So that is the drug space. Then we have the disease space, certain kind of information. So those are structured database information clubbed together with, let's say that information extracted from the literature and then joined together, and you can build a model on top of them. So yes, you can do that definitely.
Okay, thank you.
Is there any more question?
So participants may ask any question. So sir, then one it up here, sir. It was a very interactive session, and we have learned many things from whatever you have discussed today, and would like to thank you from all the Guwahati University side for accepting to be a resource person here and delivering such a wonderful talk. So would like to thank you for your time and for your patience also, because there are many issues that we have faced for the online sessions. So thank you, sir. Thank you again. And uh,
Okay, thank you for
yes,
inviting
sir.
me. Thank you for inviting
Thank you,
me. I
sir.
hope that I was able to do some justice to the. I hope the participants felt the same, that they are able to get something. Yes, I will share the PPT with the organizers, and people can look into from there, and the organizers can then share with the participants.
So thank you, sir.
Okay, thank you. Bye bye. Bye all. Have a good day. Bye.